writing. Okay, I'll say that again. Now we've just started recording. Welcome everyone to this writing workshop. It's really based on a British Academy funded project for early career research and development in Africa through writing and mentoring workshops. So there are a team of people. So you can see there myself and Andrea Mills. Andrea Mills is doing the running around and keeping us all organised. Adipo Asano at the University of Eldoret in Kenya, and Akinado Latunje, University of Ibadan, Nigeria, Christian Hartmann Institute of Research for Development, France. Can I ask that we have, if you could put yourselves on mute, please, if you're not already. So the idea is we have hubs in the UK, Kenya and Nigeria. We were aiming for a hub in Senegal, but we have dispersed that hub across a few countries, as I'll explain in a moment. So we're doing this with the help of the network of the Society for Environmental Geochemistry and Health, which I'm currently president for and hoping to hand over the reins to someone else in the next couple of months. Deepo and Akinad are also members of the international board representing Africa. And if I go through, I'll give you a quick just background to the program. So we're trying to connect environmental sciences to outcomes for human health and social economic well-being. That was one of the drivers for the funding from the British Academy. So what this will do is to help develop capacities among African researchers to produce international research papers. Now we've got more people on the group than we expected, so we may have to broaden the interest there rather than just papers to other written outputs. We're also aiming to provide some mentorship, and we have a couple of journal editors, and our first speaker is an editor-in-chief for Chemistry Africa. Andrea, can you mute those people, please?
There's a few people I can't mute, Michael. I don't know why.
Okay, can everyone please mute? Okay, so what we have aimed to do is engage willing early career researchers in subsequent peer review. So that's so you get a practice at peer review alongside mentors beyond this project as well. And with the SEGH, we do have a mentorship program where we are linking ECRs and experienced people together, and also giving ECRs experience in peer review for the Society Journal Environmental Geochemistry and Health. We're going to try and give exposure to various writing skills for different formats of science communication. So this could be web blogs. Could I ask Ebenezer, Kudamnia, please mute. Ebenezer. We're also going to be looking at different styles of writing, such as short briefs for management policymakers. So we've got a variety of different styles of writing. Overall, to improve profile and track record. So we had 56 people from these countries: Nigeria, Kenya, Senegal, Niger, Djibouti, Malawi, and Zambia. We've dropped to 53, partly because we set a task back in July to write a blog of less than 500 words to a general scientific audience of a topic of their interest. 3 or 4 people dropped out and decided not to do that. So what we've got is an idea of writing skills and where to start from, partly because we have a very broad mix of experience from student to postdoc work, participants from universities, research institutes, from natural resources to agriculture. So we're just going to widen the type of writing skills because of that, and the blogs were a good place to start and will form the basis of our group activities this afternoon, which will be a closed workshop for this cohort. So we undertook a survey at the beginning of this project back in June, and a lot of common feedback was to improve report peer review writing for career enhancement. So there's a recognition that good writing skills is good for career enhancement, and also involvement in projects, whether that's in their own country or internationally, and to develop personal networks. Now, there's clearly demand for this kind of interaction. We were aiming for 35 people and thought we may struggle to get to that, and we had to cut it off. We had a very quick recruitment exercise, and we ended up with over 50 people. So yeah, clearly there's demand. So for the morning open session, we have 2 talks, which we've made open to other societies. So we have Dr. Mohamed Chehimi from CNRS. He's also an editor-in-chief for Chemistry Africa, which is a new, vibrant and exciting journal, which has been around, I think, for 2 or 3 years, which I'm sure he'll say. So do have a look at that journal. And then, so that will be his experience on how to write a scientific paper. And then the second talk will be from myself and Christian Hartmann on the value of quality measurements for scientific writing. That's one particular, let's say, annoyance or that I have when I see papers for peer review is perhaps the lacking in quality control for measurements in scientific papers and interpretations. And then the afternoon session is a closed session for group activities. This will be run by Adipo Sano and Akinad Olatunji. So if I stop sharing, perhaps we can move on with the next stage. If everyone's okay, are we all on mute? I think we have a couple of people not muting. Please do put yourselves on mute. Just make sure the experience for others is okay then. So over to the first talk, please. Are you ready to go, Mohamed?
Yeah. Do you hear me?
Yes, loud and clear.
Oh, okay. Let me share the screen. How do I switch to full screen?
You have an icon at the bottom, just next to where it says 100. No, just where it says 100. That's it, that one.
Perfect.
Yep.
Okay.
Yeah.
And the top menu of Zoom, how can I remove it? No, not possible. Okay, it does not matter.
Yeah, you have a gallery at the side. You can just minimize it.
Yeah.
Icons at the top of the gallery.
Yeah.
Otherwise, you can leave it there. That's fine.
It's okay. It's okay. Okay, thank you very much. Good morning, everyone. I don't know if it's good morning or good evening. Probably some people are from India. I asked one of my future postdoc to connect. Thank you very much, Michael. I think it's a challenging task today because I've never done this in a workshop. I gave some lecture to young students, young research students, and early career lecturer and researcher in France, in Qatar, in Doha, and in Tunisia as well. So I'm pleased to share my what I suppose to be my knowledge in writing papers, and of course, whatsoever I give here as advice, you take it or you make it your own way. You learn from it, and then you go your own way, and you personalize, you customize your way of writing. Okay, so I don't know why it does not work. Michael, I have a problem. It does not shift. Oh, okay. So a brief summary of my career. I'm research director in CNRS France. I got my PhD in 1988, and then a habilitation in science, which is equivalent to a DSc. and my research interest is in surface chemistry and surface analysis. I published over 330 papers, and I have over 12,000 citations, and this is mostly due to numerous collaborations. And I know, Michael, you collaborate with many African countries, and I serve now as executive editor-in-chief of Chemistry Africa. The journal has started in 1st January 2018. So 4 years ago. Let me show you this. In Morocco, Algeria, Tunisia, to pass a PhD, the students have to write at least 150 pages thesis, and must publish, this is mandatory, at least one article in indexed journal. In Senegal, also there are publications are not mandatory, but recommended. In Belgium, there are two vivas, one in private with the external reviewers, and one in public 1 month after. And usually the publications are recommended, not mandatory, but recommended. In Sweden, for example, the students must publish at least 4 or 5 peer-reviewed articles, and they summarize the thesis within 50 pages. So the most important, for example, in Sweden, is the peer-reviewed articles. So you can see for either France, Belgium, Sweden, African countries, peer-reviewed publications are common to most countries. And this is why it is important to learn how to write research articles. So let's start from the conceptualization of the research. And what do you have to do once you get the first results? How to shape the future article, how to sort out the list of figures, tables, and you start your writing up. For me, the figures and tables, is the article scaffold. You will build your text around the figures and tables, and then after the writing up and editing the paper, you have to submit with a cover letter, to a graphical abstract, and any supporting information if needed. And there comes the revision. I will talk, if I have time, on plagiarism, and the publication and dissemination. The work now, I see more and more. I mean, the papers that are submitted, they are mostly tackling social economic outputs. What I see in the domain of chemistry, environment, and water quality, new power sources, biomedicine for theranostics, biosensors, pharmaceutical. For polymers, we talk about anti-fouling, bio-based biodegradable polymers, 3D printing, and so on. So you have the idea, or you discuss your idea with your supervisor. I mean, I'm talking to the young students. Usually you just need a pen and a sheet of paper, and then you draw some schemes, and you make a decision which directions to go to. And then, of course, the research you're going to develop, it needs some, for example, in chemistry, I give the example of chemistry. You have to sort out the list of available reactants, the list of available facilities for the characterization, or to find, for example, collaborating laboratories who have the facility you don't have in your own laboratory. And then, of course, you have to purchase missing chemicals and plan and schedule experiments. So if you are lucky enough, after a few weeks, and you have first publishable papers, of course, you have to write, as Michael says, usually a nice story around your results. If you have sufficient number of results that could make the object of a paper, you have to select a journal, and of course the criteria for journal selection is relevance to the domain where you work, the visibility, probably the impact factor of the journal. You want your paper to be published in high impact factor journal, but sometimes it's difficult. And sometimes low impact factor journals are even more difficult because they are, for example, very focused journals written by and for experts in one given domain. So sometimes do not believe that low impact factor means easier to publish. It does not mean that at all. So the communication of the results, the way to write the article depends really on the choice of a journal. You will not, if you have a paper on composite materials, you will not write it the same way when you send it to a composite journal, or if you have surface chemistry or surface analytical aspects of the composites, you will not write it the same way when it is intended for a surface journal. Okay, so this is the outline of generally of a paper. You have the article title, the correspondent and authors and affiliation, the introduction, experimental, or in some journals it is called materials and methods, results and discussion, supplementary materials, of course, acknowledgement for financial support, and the references. For example, if you write for American Chemical Society a regular paper, the maximum allowed is 60 references, and you are allowed a maximum of 25% of self-citation. Now, if you when you think about your paper, you have to write your paper. For me, what is important is to set the list of figures. If you set the list of figures in a logical order, you will be able to easily write your paper, because you have the list of figures. You have to describe these figures, tell what these figures are showing, and what is the story about behind every figure. So, and you have to remember that every picture tells a story. And from that, if you have, for example, 5, 7, 10 figures in a paper, maybe 10 is maximum. For example, American Chemical Society allows a maximum of 10 figures and tables. You will see that you have already written beyond more than 10, 15 article page. So that you have the tables as well to report numerical results. The step 2, discuss the results. You have to compare to the literature your finding. And then when you will come to the conclusion, you have to summarize the findings and their interpretation. And of course, you have to tell what is significant, what is new, and which is contributing to the field. Contributing something new, of course, to the field. The paper title must be short and eye-catchy. I've seen from the blogs one eye-catchy title about the hidden hunger in Malawi. I like it very much, that title. And of course, if you write the abstract at the end, when you shape your paper, probably then you will be able to write a short and concise abstract with salient features of the work and their importance. Sometimes it slows down, I don't know. Something wrong, Michael, I'm sorry. I don't know why it slows down.
It's okay, we've got plenty of time. So.
Oh, okay. So as I said, the title must be short, concise, eye-catchy, but sometimes it is not accepted. One day I wrote a paper in 2013, it has been published, and I wrote diazonium cation exchange clay off the beaten track method for making nanocomposites. And the editor did not like that, and he said to me, please consider a different title for the manuscript. Off the beaten track is a phrase that might not make sense to all readers. So I changed the title, but I found it less striking, like an efficient and unfrequented route for making nanocomposites. So the title can be changed, maybe because it is missing a keyword, maybe it is too long, maybe it is not eye-catchy, maybe it is too complex, or maybe it has some phrasing that is not accessible to everyone. So the title also can be revised after the as requested by reviewers or editors. So the abstract, you have the what is called in journals of medicine or dental science, dental medicine or biology, they are called structured abstracts. So you have to write the title of each section: aims, materials and methods, results, significance. So you have to the aim of the and the objective of the paper are usually it's a short sentence, then you describe the method, then the results you have obtained, and then you talk about the significance. In a more scientific journal like chemistry and physics, there are no structured abstract, but you can see I have here written in different colors the different part of the abstract of a paper I have published a few years ago with a Tunisian PhD student, but you recognize the same sections. We have the general context of the study, the objective, the scientific objective, the method, the results, and the significance of the work, and with a total of 224 words. And I learned from this workshop that some blogs where Michael, you asked, I think, to write some abstract with a maximum of 200 words. Some journals of, for example, from Wiley in polymer chemistry, on top of this abstract, they ask to put in a small frame in the front page of the paper, a very short description of the work and its objectives and the main results. So you have to learn to stick to the guidelines of the journal, and sometimes you have, for example, for short communication, very short abstract within 50 words. So you have to tell your story, a very catchy story within 50 words, and this is it's a difficult task, and you will see that there are a lot of words that are sometimes useless, and when if you want to strike your abstract to be striking, you must tell the whole story with a minimum number of words. So this is the okay, for the introduction. We have to talk about the context of the study, the scientific problematic, the methods reported in the literature, the problems, limitation encountered in the current literature, the problem-solving strategy you propose to address an important problematic in your domain. And usually, as an editor, I frequently see a lack of novelty statements. So the people summarize the finding of the literature, and they say, in this paper, we have done this and that. This is not accepted by editors and reviewers, because there is a short sentence, maybe one or two sentences between the upper part of the introduction and the aim of the paper, where something is missing. So you have to tell what is new and worth to be reported, and what is your contribution. How do you solve the problem? How do you solve the problem? If the problem is important, of course. So, for example, it could be for next time. A problem for the exercise for the workshop is to read an introduction and identify all these sections. So this is very important. So, for example, I took the blog of Malawi's Hidden Hunger by Ivy, I don't know the pronunciation, Ligo, and you can see in this introduction that there is the general context here in black in sub-Saharan Africa, micronutrient deficiencies are common in poor and rural areas, and so on. And then in Malawi, so here it is more specialized to, more specific to Malawi. And here you can see in another section the problem to solve, the approach proposed by the researcher, and then the suggestion to use selenium and iodine to use them to solve this problem of nutrition in Malawi. So if you read this introduction, you can split it in different sections, and you can see the logical sequence of writing up the introduction. And this is common to all fields actually. It could be chemistry, physics, geoscience, and so on. And this is the same. I took this from a recent paper I have published. You can see here in this blog, there are no references. Of course, in a regular research paper, you need references. Every statement, for every important statement, you have to give references. You have to introduce slowly your topic. Here, for example, it was on olive stones as agro-waste for the making of catalytic nanocomposites. Why we used this agro-waste from Tunisia, because it's among the top 5 producers of olive oil by volume, so it is abundant agro-waste. And we have talked in this paper about the originality, what is new in the how we combined previous methods of making supported nanocatalyst with activated carbon from olive stone. So somehow you have to show that the work is new and it is worth to be published, but you have to convince of course the editors and reviewers. Of course, I think Michael will talk about at length about the experimental section and the accuracy in getting the data, reproducibility, and so on. But please remember that even the supplier of a chemical is important. I remember my friend Stephen Arms from at that time, he was in Brighton, and he would try to reproduce some colloidal particles described in a French paper by a very well-known team, by I think I can't remember, Professor Garnier, and he couldn't. The results were not reproducible. He suspected maybe some contaminants in the chemical he used for making these colloids, and he has checked that the supplier was at the time Ron Poulenc. So he even purchased the chemical from France, from the French supplier, and to try to reproduce the results. So everything in the experimental section is really important. So you have to tell from where are the chemicals, the instruments you have. I remember one of the first papers that Michael has reviewed for Chemistry Africa. He asked the details about the instruments used for the conducting the measurements in the domain of geosciences. So anything must be clear. Of course, if you use concentration, you say, for example, I used a solution of 1 mole per liter. 1 mole per liter does not mean everything. It means you have 1 mole, you have 1 liter. But did you use 1 liter or did you use 20 centiliter? So it is, or 20 milliliters. So it is important also to indicate what is the exact mass and the exact volume used, and not only give the concentration, which is maybe the student cannot know if he must prepare 1 liter or only 20 milliliter solution. So for the main section, results and discussion, you have to, of course, to organize it, maybe to split it in subsections like here, 3.1, 3.2, etc. Start with the general strategy of the work. Maybe it gives the workflow and the sequence of the steps in designing materials or in conducting on-site measurements for soil pollution. And of course, in the domain of chemistry, if there are any computational aspects, usually they are done at the end to explain what is going on in the chemical reaction or in the interactions of materials and so on. And of course, it is essential to discuss the results in the light of the most recent relevant literature. If possible, if you have performances, if you determine performances of your materials, it is also now common to provide a table comparing the performances of your materials to those similar materials reported in the literature. So this is the outline how you can break the results and discussion in different sections. Now, for example, for the strategy of the work, usually now we make a lot of complicated materials, multi-component materials, and usually I recommend that, for example, for chemistry, that you describe the way you have designed your materials. It gives a clearer idea what you have done in your work. We have done this once in results and discussion. The reviewer preferred that it is displayed in the introduction, so we had to revise and to shift this kind of cartoon to the introduction. Comparison of performances, of course, you have to every results you have, every plot you display must be discussed and of course interpreted and discussed in the light of what is reported in the literature. And for performances, we can see here the kind of materials, the experimental conditions, because maybe you can have, apparently you have lower performances, but we have to know if you worked in the same experimental conditions or not. So you have the experimental conditions. For example, here it is a degradation of a pollutant, the initial methyl orange concentration, the rate constant of degradation of the organic dye. And here are the references. We have taken here the materials that are mostly close to the materials we have prepared. And here you have your work and you compare to what is done in the literature. Okay, so when you present the figures, for example, you can see here, I see this very frequently in Chemistry Africa, figure 2C. Now you have to have figure 2 with A, B, C if you have 3 figures. So the C must be inside the frame or slightly outside. Like in here, this is accepted. We have a figure with 6 plots, A, B, C, D, F. This is correct. This is not correct, and we had to ask the author to change that. For example, you have to save space in your article to make it more concise. For example, here is surface analysis by X-ray photoelectron spectroscopy. We have 3 materials, 3 survey scans. And here we have the narrow scans, high resolution narrow scans. You can put them in another frame, but sometimes this is sufficient, and you can have here the high resolution scans as insert in the figure. Instead of making a table with for example, the atomic percentages here, we can a bar graph sometimes is better, because you can at a glance here, in a glance, you can see the differences between the materials instead of reading numbers in a table. Another way to present your data, you have, for example, spectra and the composition here as a bar graph. So this is it makes I see this many times in American Chemical Society papers. So I found this interesting, and I published this with my colleagues from Sharjah University. Take digital pics as much as you can. Now it is many papers they show. For example, here it is a catalyzed degradation of nitrophenol. We start from colored samples, and it is discolored at the end of the catalyzed reaction. So this is the reduced form of nitrophenol, and you can, this is interesting for the reader, for the newcomer in the field. So combining the plots, spectra with digital photographs is really interesting for the reader. So for the conclusion here, you can see this is one block conclusion, but if you read very well the conclusion, you can find the maybe some reviewers, they ask you to recall in one short sentence the objective of the primary objective of the work. You summarize the essential finding. You have the conclusions, your conclusions must be sound and justified. It means that you have justified the conclusion with experimental work. Is it justified? Are the conclusions supported by experimental data? This is what reviewers and editors ask for. So what is new? What have you achieved? What are the performances you obtained for your materials? And also what I request now from authors is that they finish by a take-home message to tell what is significant what is the significance of the work, and what are the horizons it opens for the future. For now, if you, I still have time, Michael.
Yes, 10 minutes. Could I?
Okay.
Yeah, Simon.
Okay.
Simon's just muted himself, so you shouldn't have any more background noise.
Okay, so of course, when you submit, you need a cover letter. Now you have edited your paper, you need the cover letter, graphical abstract, highlights, maybe for if you publish in Elsevier, the manuscript, supporting information. If you have a video, you can now post a video as supplementary material, and list of suggested reviewers, and the reason why you select these reviewers. You have to write as if same as for your paper. You have to tell the editor why you submit your paper to the journal and how it fits the scope of the journal. You have to summarize the work, and you have to tell the editor why you're, in a few sentences or you can give highlights of your paper to tell the editor what makes your paper important and unique. For example, here we wrote to justify the publication, it will enrich the database of papers on biochar published by Applied Sciences, 35 papers only, none in 2021, and especially those on biochar catalysis combination, etc. Maybe you need to finish by, please note that the manuscript is not under consideration for publication in another peer-reviewed journal at the time of submission. Please, if you submit your own paper, we receive so many things like this. According to my knowledge, this work is real and important. Only one sentence in the cover letter. The article is original. I have seen also this research will help researchers in the field of corrosion. I also have received something like this. Sir, I am working on in college. Data is also good and extensively done at laboratory. Kindly accept manuscript. Suggested reviewer, for example, here, I think he can. I can't say he's very talented. She's honest. He's honest and talented. She's brilliant, doctor. This does not give information to the editor why he or she should invite the suggested reviewer. You have to tell why you suggest such a reviewer, because he's expert, for example, in supported catalyst, or in a catalyst for degradation of pollutants. You have to give one sentence why the exact reason to show that he's expert in the domain. For the graphical abstract, it must not be very complicated. One, at a glance, you see here the purpose of your paper in a short in a small photograph. And the photograph, I see so many graphical abstract that are so complicated. If you reduce, because it will be reduced by the publisher when it is published, you still must see it very clearly, like this good graphical abstract. But this one is not good because this one is too big. When it is reduced by the publisher on the website, for example, these details you do not see them anymore. So you miss the information. So this is not a good graphical abstract. You select the journal. For example, I gave the Journal of Applied Surface Science. If then you have the feedback from the editor about minor or major revision, you can consider, you have to consider all comments. You have to give a response to a specific comment. You have to address the comments point by point. You have to avoid replying, it's known fact or done, this is done, but you do not tell where you have revised your paper. Or people say it has been fixed, yes, but we don't know where in the paper, how you have revised the paper. Or you have to, some they say thank you, but it is outside the scope of the paper. This is not acceptable at all. You have to distinguish also when you revise a paper, when you have comments on your paper, between should be and mandatory. Should be is not really mandatory, but you have to discuss anyway. Mandatory, sometimes an experiment is, you have to repeat an experiment or to add new experimental work. That's, without that, the paper will be rejected. We can also see the paper lacks originality. And for example, you have to tell, even though you have said it in the original submission, you have to explain what makes your work original. So for example, here we reply to this comment: the research contents of this manuscript have no enough novelty to catch readers' attention. So we have replied, we have stressed in the introduction what it is new and worth to be reported. But we highlight, of course, in the revised version, the part of the paper of the introduction of the paper that shows that the work is original and is worth to be published. Okay, I skipped this one. So how to make an impact with your research? Of course, we need good science, original, challenging. You have to explain the context, the objectives, address open questions and challenges, provide your strategies for problem solving. Your paper should not seem to the reader's eyes that it is only incremental science, that you are just adding a new work but without any breakthrough. It must add something important to the field. You have to write novelty statement in the introductions to attract your future readers, thoroughly discuss the results, and always give a take-home message in the conclusion. You can find this blog written by the the founder of the journal, Elsevier journal Carbon, Peter Thrower. 8 reasons I rejected your article. You will find it easily in the internet. So he gave the 8 reasons. It fails the technical screening. You do not follow the guidelines. It does not fall within the aims and scope of the journal. It's incomplete. The procedure and analysis of the data is seen to be defective. So this is the aim of the next talk. Conclusions cannot be justified. It's simply a small extension of different papers. This is what I've just said to you. It's incomprehensible. It is boring. Also, you can see in this one, in the first item, plagiarism. So I can show you here problem of plagiarism. Here you can see paper screened by Authenticate software. You see that it is copied verbatim from different papers. And the duplication of the results, you can see here, this is published previously, and you can see the spots here. The author changed the color. This is SEM. You can see here, this is the same. This is the same here. This is here the same. And here is the same. So this is duplication of results, and the paper has been rejected. So this, you have to be very careful with this. If you reproduce a figure, you have to credit the source, and you have to explain why you reproduce the same figure again, maybe for a new comparison. Also, we reject papers for the following reasons in Chemistry Africa. Authors do not respond to all reviewers' comment. Reviewer 3, the authors did not take into consideration most of the remarks and suggestions. So this is a reason for rejecting people. Rejecting the articles, sorry. And now, after Peter Thrower, 8 reasons I rejected your paper, these 2 guys, they wrote the opposite way. 8 reasons I accepted your paper. So the first one, I cannot see it. Oh, okay. It provides insight into an important issue. The insight is useful. The insight is used to develop a framework or theory. The insight stimulates new important questions, and so on. The articles tells a good story. This is what I see most of the time from the comments of Michael Watts when he serves as editor for Chemistry Africa. Okay, so now you have published. What is next? Of course, you have to valorize your work, present work in conferences. Oral presentations are more attractive, better than poster, but poster you can have more time to discuss your findings. Invest in renowned international conferences if there are some financial support, of course. Write review papers and book chapters. This is very important to show that you master a given topic. Share your, of course, everyone now shares results on social media. Create ORCID account. And of course, if you are a renowned author, good reviewer, next step is to be so next step is to become editor. I skip this. So whatever you do, you cannot force people to cite you or to recognize your work. And even though you write a science or nature paper, you can see here that analytical biochemistry is not a very well-known journal. But you can see that Marion Bradford in 1976, she wrote a very nice paper on the new method to determine protein concentration in solutions. And her paper has been cited up to now over 235 6,000 times, and you can buy actually the Bradford reagent from many suppliers. So whatever you do, you do a very good science, very challenging. It is a breakthrough in any domain. Now getting cited remains very difficult, and you never know. The more you write, the better it is. The easiest it will be for you to write papers, reviews, chapters. So as a take-home message for young researchers in early career, students, constantly survey the literature, receive alerts possible for updates and new articles in your domain. If you have an idea, you have to try it, of course. Write articles before getting complete set of results. It saves time. I learned this from John Watson when I was his postdoc in Surrey University. PhD students should publish most articles of their articles before the PhD viva. After the PhD viva, maybe you have a job somewhere else, you change countries, it will be more difficult. A good thesis for me, at least 3 papers, 1 per chapter, 1 review or a book chapter. So the literature survey of your thesis should be published afterwards in a review or a book chapter. This is possible if a student is guided by a senior and understand the structure of research and review papers. It will be possible to publish during the series if, of course, you have sufficient number of results. And the objective, you start your PhD or you start your in early career, you read a lot. What is important is to change your status. You shift from a reader to an author, and the more you write, the more you will screen very quickly the literature. The more you learn, and the more you will discuss, compare your results, and write and write and write. And I would like to finish with this picture of William Turner, the father of Impressionism, British painter. He inspired Claude Monet in France with his painting Impression le soleil levant. Why William Turner? Because William Turner has done over 19,000 painting and sketches, drafts, and so on. So I think he was painting all day. So if coming to science, the more you write, the more you do, the more you will inspire people. And this is very, very, very important to learn to write your papers and disseminate your knowledge. Thank you very much, and thank you for your attention. Thank you, Michael. Hello.
Thank you very much, Mohamed. That was a wonderful presentation. Perfect timing, just on what I was planning before. So that's wonderful. Many thanks for that. And I'm sure everyone's very, very grateful for your insight and advice. Could I perhaps open up some questions? So you could perhaps put something in the chat box or put your Zoom hand up. So we have a comment from Manikos Dimitriadis. What about considering also keywords?
Oh,
yeah.
Sorry. Yes. Oh, okay. Yeah. Keywords. Well, it depends on the journal. 5, 6 keywords, sometimes 4. And an example, I don't know. Let me. Okay, so for example, last time, the last paper I have published, it was on supported catalyst. We wrote, if I remember, because I don't want to change screen, olive pit, that's the biomass, biochar, because we pyrolyze the biomass, catalyst, methyl orange, probably depollution or something like that. So the keywords must also tell the whole story of your, the main important parts of your work.
Right.
So I suppose for soil pollution, it must be the kind of metals, or maybe the series of metal, the soil, where it is, the kind of soil, the country, maybe.
Okay, wonderful. You're getting lots of comments, and thanks for your insight and advice. I don't see any other any other questions at the moment. If I'm missing you, please just shout out. I think maybe they're a little bit shy at the moment. So maybe we can save some questions for the end of this session, perhaps, if there's nothing else right now.
Yeah, okay. I'm open to
Correct.
any. So maybe they can also, if I can be of any help, they can send me an email to my Yahoo email address.
Wonderful, thank you. We do just have one more question from Deepo Sano. Does the quality of the article that you cite, does that matter?
Does the quality of the article?
The quality of the article, does that matter?
Of course.
The article that you cite.
Yes, yes. It's, I mean, it's the most difficult. It's very difficult. I didn't show you my folder here from my computer, but we have up to 10, 12, 15 versions of the article before it is sent out for submission. So the more you edit, the better it is, but you should not at a certain time, maybe you cannot see the defects, the things that are lacking, and sometimes you have to go to hope for the best and submit your paper. But of course, the quality, first of all, the quality of the data, the quality of the ideas, the quality of the research, of the results obtained. Of course, you gather all this and you write the best you could.
Thank you.
Yeah, quality is of course, the most important.
Just to pick up on one of your points, it's important just to have a go.
Is it important to
just have a go at writing and just start? Often people that I've managed, it's the fear of writing that stops them.
No. You have to, the more you write, because when you write, also when it is difficult to start to write, well, because you can read 1,000 papers for the literature survey, but try to write a summary of this finding, even in one page, what you have learned from these from the literature survey. Well, it's difficult. But if you do not start, if you do not even, if you do not start and if you do not use your own words to construct the text, it will be always difficult. Once you start, it will be difficult. I always say to my student, the first paper you will put 6 months to write it. The second one, maybe 3 months, maybe, and then maybe 3 days. And if you are experienced, in one day, you can have the first draft, and then you have to add the results and the discussion and so on. So, but the first one will be, of course, difficult. And it is also difficult when you change subject. For example, I started to work on biochar. It took me ages to write my first paper on biochar because it's a new domain. I had to learn something new. I have to learn the major aspects of making activated carbon and biochar. So it took me time, and I had to learn as well as a newcomer in the field, and as a student, almost as a research student.
Okay, great. And I think however many papers you've written, it is always useful to have other people read it. I often get my students to read what I've written, and they will come up with things and mistakes that I've made. So
of course,
there is always an opportunity to improve and recraft.
Yes, and the last thing I would like to say, Michael, is the first time you write, of course you are an author, you write, but with a little bit of experience, the more you write, the more you shift from author to reviewer. You have to become at a certain time your own reviewer. When you finish your paper, when you edit the paper very well, you have finished your work of author. You have to sit back and look at your paper with different eyes. And the first person to read your paper is the editor. How your future editor will see your paper. So this is one thing. Suppose now you are lucky and it passes the filter of the editor and it comes within the hands of the reviewer. You have to read your paper as your future reviewers, and you have to find what are the weaknesses in your paper, and you have to improve your paper that way. So you write your paper as an author, and before you submit it, you change your status from author to editor and reviewer. And this is how you improve your writing up.
Yeah, experience as a reviewer definitely helps your ability to write. So
I removed that. I'm sorry, I removed that from my talk.
Well, that's partly what we're doing with the group activities this afternoon. So there is method behind it. So we have a couple more questions. What is the minimum number of pages required for a publication?
If it is, usually if it is 1.5 or double spaced, usually journals request 25, 30, between 20 and 30 pages, included tables and figures. If it is short communication, you have a very strong new studies that is worth to be reported in short communication. I think you have to stick to the guidelines of the journal. For example, if you publish in suppose in chemical communication from Royal Society of Chemistry, and you use the template of the journal, it must fit into 3 pages only, and anything else can be shifted to the supplementary to the supporting information. So 3 pages. But with using the template, I don't know if you don't use the template of the journal, maybe it is 6 pages or 7 pages maximum.
Yeah. And of course, each journal has very useful guidance for authors. So do read that before you start writing your paper. Every journal, there'll be common themes, but they might be just slightly different, mightn't they, between publishing houses. We have a very big question here from Akinad Olatunji. How do we address the challenge of accessibility to quality journals, articles in reputable journals by most African students and ECRs, where often you are requested to purchase access? So most ECRs now have access to mostly predatory journals that are open access and readily available. This has led to referencing substandard papers by ECRs. Yeah, it's a big issue.
Yeah, but I mean, the university must have registrations to ACS journals. I don't know.
I certainly know from the experience of talking to people at Springer, they do have free institutional access for lower middle income countries, and often institutes don't take up that offer. So I think it's institute by institute. So your university or research institute has to apply to the journal, and then they will allow you free access. And of course, this cohort for the writing workshop will all be members of SEGH, and therefore your membership will give you access to the Environmental Geochemistry and Health Journal. So you will have access to a journal to help us through these workshops to the end of March.
Something else, you can write directly to the main author. Main authors, when they receive a request like that by email, they usually send the PDF to the person that requests
and often there are free open access research archives. So in the UK, for example, we have something for environmental sciences, the NERC Open Research Archive. It's NORA. So many papers, the original Word document pre-publication can be saved into there.
I didn't want to talk about it.
Yeah, for certain publishing houses. Not every publishing house has an agreement.
Yeah.
Yeah.
I didn't talk about the preprints and preprint service. There is for your domain, there is I think Earth Archive, something like that.
Okay. I think this is almost another topic. So these are interesting questions that are coming out. Christian has his hand up.
Yeah, I just wanted to make a comment about the access to high-ranked journals that you have to pay. There are also programs, I don't remember if it's United Nations or UNESCO, and in soil science, for example, for me, or agronomy, it's Research for Life, and the library in as you said, middle or low income countries can have access for free. And very often the researchers of the libraries do not try to get it when everything is prepared for this. So perhaps there should be a special session during the training about how to access the papers, because it's a very important question.
Right, I think that's it for
Okay.
questions. Many thanks. So we'll do the next presentation. I'm sure people may think of more questions that we can go through at the end of this session for discussion. What would be quite useful is if experienced writers on this call, is perhaps they could put some of their top recommendations in the chat box, and maybe we'll end up with another list. And I'm sure with the wealth of experience, you know, everyone has a slightly different perspective and experience. We might build up some additional tips, because there are many other things. I'm sure that you know it's time limited.
Yeah.
We could probably discuss this for hours, couldn't we?
Yeah.
Okay. So could you unshare your presentation, please?
Thank you.
Many thanks.
Thank you. Thank you. Thank you.
Very much appreciated. So Christian, you have the next article saved, the latest version from us. Sorry, the next presentation. Okay. Great, thanks very much, Christian. So, so we're going to split this presentation in two. So I will start out with some examples of why quality measurements are important and the decision making process of that. So you may work with GIS, you may work in statistics, and you may put all your effort into really nice, elegant statistical interpretations. And often, as I see in papers that come through for review, the measurements are not ignored. So I often push them back and say, well, if you haven't got confidence in the basic measurements at the beginning, or even in the sampling strategy, how can you expend all of your effort in some really elegant statistical interpretations? So I'll give some examples of where this may be useful, and then Christian's going to go through and actually explain this doesn't have to be difficult or expensive. Thank you. So the first slide, many important decisions are based on results of your field. So you almost need to think of the quality of measurements in the field, not just the laboratory measurements. So how can you be sure that your field measurements are okay? So I've been on field work with a very senior professor in analytical chemistry. Having seen some field data from this person, I was wondering why the conductivities were 0 or close to 0 all the time. And when I was in the field, he realized he had the cap on the probe. And that's why, of course, he was getting 0 or close to 0 for his conductivity. So that can happen to the best of us, is how do we make sure that we have confidence in our data? And it's very important for the interpretation. It is important to have indications of the quality of your results. Often for publication so that we can be sure that the work is reproducible. But also, if you're presenting data to a client, you need to develop confidence or trust. I know there is an issue in many countries where commercial companies, big mining companies, for example, will automatically send samples to Canada or to Australia, to the big commercial labs, because they won't trust local laboratories and the data. It doesn't have to be like that. And results may be used to make a regulatory decision, a commercial or a scientific decision. Now, all of the best labs around the world can make mistakes, so don't worry about that. All labs make mistakes. What they must have is a process of monitoring and evaluating and learning from those mistakes. So there are no labs that are perfect, okay? So that's the thing just to remember. And when you are sending your labs off to Canada or Australia, don't assume that data is perfect coming back and going into your papers. You will need to write in your papers how that data has been produced and how they provide the confidence in those measurements. Just writing it was produced in a lab in Germany, or it was produced in France or the UK, that won't be enough. So increasingly, as we're seeing with globalization, increasing competition for research funds, we're seeing increasing interconnected work across the world, and of course Zoom is rapidly increasing these collaborative opportunities. We need to remember some of these sort of principles to enable reproducible data and reproducible experiments. So how are we going to demonstrate the quality of the results? And to demonstrate the results can be compared to results produced by other laboratories or for comparison with other scientific literature. So it's all about being able to compare and reproducibility. Could we have the next slide, please? So, of course, as I've just said, everybody can measure, but not everybody can do professional quality measurements. And remember, even highly creditable labs can make mistakes. Even labs with accreditation make mistakes. Official ISO accreditation just means they are doing what they said they were going to do. It does not guarantee the quality of the measurements. Of course, it does help that they will have a system in place to monitor, evaluate, and learn, but you do not need ISO accreditation for that. There are much cheaper solutions for that may be more appropriate to your local needs, perhaps. So a lot of these ISO standards are generated by people in Europe, for example, with access to modern resources and equipment. So I'll give you some examples of why measurements are important. So many of you will be working across food security or food safety, contaminant exposure, or you may be working to develop tools for policy advice. And if you can just have the next bit. And some of those key components for this group may cover analytical chemistry outputs. It could be testing geospatial control, so you may be working a lot with GIS or geostatistics tools, modeling, predictive modeling type tools, and increasingly machine learning. And of course, experimentation with lab field trials, and there may be some degree of integration of different layers of data, particularly with some of this cohort where you may work across environmental, geochemistry, and health, and linking those, making those interdisciplinary links. Okay, can we have the next slide, please? So we have an example of an integrated data set here. So this is from the geochemical baseline of England and Wales, where you will be pulling data on a large scale, so on a national scale. This will be over 25 years. So of course you have your challenges in ensuring the measurements of data over that period of time, because technology changes, instruments are changed, sensitivity detection capabilities of instruments change. So you need a reliable means of ensuring the consistency of data, particularly over long periods of time. For experiments, you may be running experiments for a number of years. There could be small experiments, there could be large baseline type surveys. But equally, this applies to small scale studies. Okay, next slide, please. So there's the research cycle. I often use this for a laboratory setting. So you could be at any point in that cycle. You could be at the study design and commissioning laboratory measurements. You may not be doing the measurements. There'll be specialist type data, or you may be at the point of designing and the predictive models and risk models or maps. Or you could be at the other end interpreting advice, providing advice and publishing, or at the policy end. But all the way through that, you need to be able to have confidence in the measurements for the laboratory analysis. So really, that comes back to study design right from the beginning, is working quality into the sampling strategy, the measurements, the models, how are you sure the models are working, and then how are you sure that the advice and the interpretation is correct. So a wider team engagement, collaborative science is ever more important in this sort of day and age. And of course, there's always a financial implication if the quality of the data was lacking confidence. Okay, next slide, please. Okay, yeah, we can keep going, thanks. Common problems in the literature that I've come across, I think we've already seen this morning, a lack of or unclear aim and objectives to achieve that aim, lack of or no detail for quality assurance measures. So that will lead to a lack of confidence in measurements, in how the field collection strategy was designed and how the samples were collected and preserved. So preserved to maintain their integrity, so that they are that your measurements are representative of the samples at that point in time for collection. Often papers are missing that information. So if that isn't right, you could question the complex statistics and very elegant graphical representation interpretation. So often, remember, decisions are founded on data and could be costly without appropriate evidence for traceability. I think we had a question from a depot. Does it matter what the quality of the paper is that you're citing? Of course, if your paper's out there, you may cite that paper and say, yes, well, so-and-so said this, so that must mean it's okay. And then someone will come along and cite your paper and say, well, so-and-so, and then so-and-so wrote this. And then there can be an embedded error in the literature, potentially. So getting the quality at the very beginning, that's the key message. So your paper or report may be rejected without transparent and traceable data. Remember, if it's a report for a commercial sense or a policy advisor that's making a decision, there could be a legal implication for that data, not just a financial cost. So always think about that kind of repercussion. Next slide, please. So we saw that Ivy had produced a blog for hidden hunger in Malawi. So we'll give you a few examples where reliable data may be important for intervention strategies to tackle things like hidden hunger, where you have micronutrient deficiencies, which have a real human impact, where some micronutrients may have quite severe health costs, or they might be easily remedied. In Malawi, for example, this is an economic cost from about 6 or 7 years ago, I think we did as a team, for zinc deficiency in Malawi using disability adjusted life years. So this was valuing a productive life. Zinc deficiency alone was costing the economy $51 million a year, which is quite significant. So you need good data to plan intervention strategies and what is the best approach. And if you're conducting experiments to understand the best intervention strategy, whether that's giving people tablets, adding zinc to fertilizers, or spraying it onto plants, you need to know that your experiments that follow are providing quality data to advise the best approach. So if we can go to the next slide, please. I mentioned about producing very elegant maps. So a lot of data that you may produce for food samples or soils or baseline type work may end up in very elegant maps like this, where it's just showing at an African scale the dietary iodine supply. And then there may be nice, elegant calculations to then predict the risk of dietary iodine deficiency, but it all needs to be founded on good quality data, good quality measurements. Next slide, please. And as I said, there may be a range of alleviation strategies. So there could be biofortification of crops, crop breeding, and all these experiments need good data. I've seen some presentations where people have been deciding on multi-million-dollar field trials to biofortify staple crops with zinc, and you look at the chart and you see the difference in applications being presented on a bar chart in such a way that actually it looks quite significantly different. But when you look at the axes, you're looking at sort of like maybe a few % difference, which could be analytical error. And then what you must do when you're seeing charts like that is actually question the person. How do they ensure confidence in their measurements? That could be an analytical error. It could be just acceptable error and uncertainty. And actually, on that example, I did quiz the person and they didn't actually calibrate their instruments on a daily basis. So you have to wonder on the value of that data, which is informing multi-million-dollar field trials. And of course, salt iodization has been a massive program in alleviating iodine deficiency around the world. But there's often a problem in the quality of the salt and the iodine content. But of course, that needs to be measured. And if the measurements aren't reproducible, then you can't be sure whether you are catching people that are not iodizing salt enough or too much. It may be that the measurement technique's wrong, not the people that are fortifying the salt. We can get to the next slide, please. So yeah, I gave an example. You might have a biofortification strategy. So this is an example of applying selenium to crops in Malawi. So if we go to the next slide, so you may have nice response curves like that as you add selenium and the response in the grain selenium. So you need to be able to have confidence in that data, and you'll see some error bars which may present the uncertainty on those data. So that may require replicate analyses, for example. So if we go to the next slide. Sorry, can we just get back to that one? So what we may deduce from that slide is what is the appropriate amount of selenium to add to a fertilizer in this example. So there'll be a cost implication. Do you add too much or not enough? If you add too much, is there a toxic, potentially a toxic outcome? If you don't add enough, are you simply wasting your money and having no effect? So good measurements will help to decide that. So next slide, please. And data may be stored in things like we have at BGS, an app called MySoil, where a lot of data may be presented to the public. So you may have members of the public accessing data through portals like this, and I'm sure there are many examples around the world, and they won't understand the concepts of uncertainty or error. So it's very important where data is getting to members of the public to have that confidence in the measurements. And of course, you may be bringing multiple layers of data together. So again, confidence in each of those sources of data is absolutely critical. Yeah, last point there, relevance for public policy. Definitely, if you are giving advice, measurements, interpretations to public policy decision makers, they are assuming a certain level of confidence in that information. Okay, next slide, please. Okay, it's just an example. If you just click on the arrow again, an example of experimentation models. If you're, as an example of in the lab, where you may be biofortifying grass in this instant, you might be coming up with a model here. So again, understanding the different inputs into designing a model all requires measured data, of course. Okay, next slide. And okay, so we've got a typical ICP-MS instrument there for measuring elemental chemistry and an HPLC there with elemental speciation. So there will be different approaches depending on what you're measuring. So think carefully about your target analytes, your measurements. Do you need particular careful considerations? So for elemental species, you often have problems with redox conditions if you're trying to look at different forms of the element. Can you extract chemical species from a solid sample without affecting the redox conditions. So are you actually measuring a representative example, a representative sample of the original sample material? So if we can go to the next slide, please. This is an example of a WAM model, and if you need to know more about that, Belinda Kaninga is on the talk. She has been using this and finished her PhD, so she can give you more advice on how that works. I'm sure Belinda won't mind. But WAM is a very simple model whereby you feed in data. Mariam, if you could go to mute, please. Thank you. So if we just go to the next slide, it's often used for solution fractionation in soils. So you'll have speciation information, and this kind of model is often used to sort of as a risk assessment approach. But it's taking in a variety of sort of chemical parameters, and then it will produce some information that will inform risk models. So it's very important to make sure that the quality of the data going in is important, because it's like any program. Whatever, if you put rubbish in, you'll get rubbish out. Go to the next slide, please. So yes, you can. This is just a photo, really, of. I think we may have Lukundu on the call. So that's Lukundu in the middle. Yeah, this is in the Zambian copper belt. So you may have to link back to real-world situations. So this is in some sort of field experimentation. How can you translate your modeling, your data presentations back to a real world environment, and then also designing your sampling strategy into thinking, what is the model going to need? So making sure you capture all the relevant information and store the samples in the appropriate conditions to feed into models. Next slide, please. So this is another example where in the UK, we were asked to help the Public Health England understand the problem with private water supplies. So a European directive was requiring private water supplies to be monitored. We simply didn't have the data on this source of water. So private water supplies generally in rural areas. It accounts for about 1 million people in the UK. So it's quite small, but 1 million people still significant. They may be on boreholes, wells, or well, very rarely river water. Actually, it's generally springs or boreholes. And there is generally no measurement by local authorities of these water supplies before we did this. And particularly in an area, if we go to the next slide, in the southwest of England. So I'll just actually mention how we collected water. So the examples of boreholes. So they could be in a garden, in a shed, they could be under a concrete block. Sometimes farmers would cover them with manure to stop them from freezing over in the winter. So there could be a variety of private water supplies in the way that they were maintained. So if we just go to the next slide. So this was conducted in the southwest of England, Cornwall, where there is a particularly well-known problem with arsenic in private water supplies, in groundwaters, because of the geology associated with that area. And what we were showing in this survey of 500 households is that 5% were above the prescribed concentration values for arsenic, and there were other elements. So all of that kind of information feeding back to households can be quite serious. So if people have a lot of arsenic in their water, you have to be very careful in communicating that to them, but you also have to have confidence in the measurements, because that can have quite serious implications for how members of the public will respond to that information. And again, there could be opportunities for legal consequences later on if it is wrong. So quality measurements are absolutely essential, not just for the policymakers to make decisions on how to change or address this problem or monitor it better, but also human reactions to that data in the household level. Next slide, please. And of course, we went back and we did a biomonitoring survey. You may want to look at different sources and exposure routes. So you have different considerations depending on the sample matrix that you're looking at, whether that's a water, a dust, a soil. and dietary inputs. So it's not just going to be a simple solution. You have to think in that particular context. Next slide, please. So of course, geochemistry crosses boundaries across the environment, resource security, ecosystem health studies. So just make sure you know how your data is produced in whatever matrix your measurements are conducted in. Do you have confidence in the data? Would you be comfortable being questioned on the validity of your data? It can feel very personal, but plan quality measurements into your sampling strategy. It's very important to think about the consequences of your measurements. And remember the aim of the study and the objectives to achieve that aim. And I think the next slide is where Christian's going to pick up. Yeah, Tom.
Yeah, sorry, I will up there. Michael?
Excellency, here we can see the full complement of regimentation.
Have you got the, have you lost the slides?
Yeah, I wanted to put the new presentation.
Okay.
But I have a little bit problem with, I don't know why I cannot come back to Zoom.
Okay, whilst you're finding that, I'll go through a few of the questions which will save, save a bit of time at the end. So if everyone looks in the chat box, you'll see that Ariadne has put in some information about estimation of measurement uncertainty. So you've got a link there. So there are many resources that you can use. That's a great one from Eurochem. Elikos has also added in another link to information via Eurogeo surveys. And of course, Eurogeo surveys are an enormous wealth of data there for European geochemical baseline surveys. There's a couple of links there that you can follow through. How are you getting on, Christian?
Okay. So,
you ready?
Yeah.
Okay.
Okay. So how to produce good quality data will perhaps be a reminder for you or not. But before I make my presentation, I wanted to introduce myself because, as it was said, I'm a researcher and soil scientist at the French National Research Institute for Sustainable Development. So just like Michael, I'm used also to work in tropical regions. At the moment, I'm more involved with Asia, but I used to work in Africa also in the past. And at the same time, I'm making this presentation as a member of the GLOSOLAN, the Global Soil Laboratory Network, that is an initiative from the Global Soil Partnership, in which we try to have data in which you can trust, you have confidence, and that can be compared worldwide, because it's the big problem at the moment, as Michael mentioned. So if it's a reminder, I'm sorry to repeat things that you know, but if it's new information, so this means this training is useful, and feel free to ask any questions, because this is a very important issue. And also not only to ask questions, but perhaps to request additional trainings on some specific subject. I will just show different aspects, and there has been a comment, for example, on uncertainty. And so you need to calculate uncertainty via different ways. And so feel free also to ask trainings or something about some special subject. So we mentioned the difference between everyday life measures and scientific measures, because when you make a different way to explain it, but let's say that in the everyday measurement, you have just a value. If I ask you your height, 1 meter, 60 centimeters, so it's enough. But in science, you have a measure is a value plus an uncertainty, and this is very important. What I forgot to write is also you have a unit. I did not indicate it, but I think it was also mentioned by Mohamed before this, but you have to take care to clearly indicate the unit. So certain is what you have no doubt, doubt, you have confidence, and uncertain is, of course, the contrary. So also in mathematics, you can say that 170 is bigger than 160, but in the real world that science tries to describe, you cannot immediately say that 170 is bigger than 160 if you do not have knowledge, information on the uncertainty. Here I have indicated different uncertainty. Probably with plus or minus 1, 170 will be bigger, but plus or minus 5 is not sure. It depends on how you calculate the uncertainty, but I will not detail here, but this is important. And 170 plus or minus 10, you see, is a totally different picture. In fact, your two measures are the same. And this was also mentioned by Michael before. Take care about the range and take care to indicate the uncertainty. So why do we have uncertainty? Why do we have a doubt on our result? Because no instrument is perfect, and the conditions we measure can create also that doubt. And there are two important concepts, that is random and systematic errors. And here we speak about measures, but we could also speak about sampling procedures, because if you do sampling in the field, there is also a problem about your statistical model, the normal distribution or not. But again, I just mention it, I will not detail. So it's important perhaps to precise what the concept of error and good data is. If we have been able to make a meeting all together in the same room, I would have given you an egg and ask you to measure the egg. And I would ask you to write down your result, and I encourage you to do the same, even with your friends or in your lab, and to see the results that you have. And you would get, of course, different measurements, because you have the true value that in fact can never be reached, and you have the measured values. So there is also a science of measurement that is called metrology, and that tells you that all measures contain errors. A measure, so let's say it's more or less something that is close to the true value, and plus or minus the error. And you see, this is the error. But as you cannot know the true value, it's totally impossible. You cannot measure the error. You can estimate it. So all measure contains errors. Thus, a good measurement, you have the minimal error. And as I mentioned to you, there are two types of errors. There is a random or unpredictable error. For example, you have a barrette and you want to read the level. You already have a problem of menisci. You have a problem of alignment to the menisci. So if you do the measurement several times, or if you ask several people to do the measurement, you will have different values with a mean value and uncertainty, dispersion of the result. And you can express that dispersion in different way. Very often it's standard deviation that is used, but there are different ways to measure. So this is unpredictable because every time it's random. And you have also a systematic or predictable error, that is a regular error. If we have a test tube, for example, this which contains the volume that is indicated, but this is correct only at 20 degrees. So if you measure this in a non-air-conditioned lab in tropical areas, perhaps you will have 35 degrees and the volume will be incorrect. If you are working in Nepal in winter and you have no heating and your temperature in the room will be lower, and you will have a different, in fact, volume, and you will have different results also. And this is a bias, a systematic difference according to the true or consensus or reference value. And in this case, we can also have two important concepts, that is accuracy and precision. In fact, in the first target, the results are not accurate and not precise. None of your hit, none of your measurement is close to the true value. And in the second case, this is precise. Every time you measure something, you have the same result. But unfortunately, your result is not accurate. There is always a bias and you cannot reach the middle of the target. And so the last one is your results that are precise and accurate. And this is always something that you must keep in mind, that you need precise and accurate results. And your measures will always contain errors. You cannot avoid it. You have seen what the errors are. You will always have errors. So you have to limit the errors to a minimum value, detect the errors that exceed the limits that you give yourself, because as you have seen, sometimes you need a conclusion about the health of the people. So you have to have very accurate and precise results, and the limit must be narrow. Sometimes you can have much larger range of acceptable errors. So it depends also on what you want to do. But if you detect excessive errors or mistakes, take action when they exist. And okay. So this is a very important concept to keep in mind: random and systematic errors, precision and accuracy, that you evaluate the uncertainty on statistical analysis that can be done in a different way, but most often it's a standard deviation. And it's exactly the same for survey. I will not detail, but it's the same for survey, and we could discuss it. And so as error and mistakes, mistakes is also when you make a mishandling. Error is when you can never know what the result is. Mistake is when there is something that you could do correctly and that is made incorrectly, and this cannot be avoided. So you need procedures to measure the errors and detect the mistakes. And then you need to take action to reduce errors and to correct the mistake. And this is very important. It's not only necessary to know that there is something going wrong. After this, you have to take action. You have seen what Michael said, there is a circle. And in this case also, you detect and you take action and you measure again. And if everything is all right, okay, you continue. If it's still not all right, you continue to check again. And okay, this is quite theoretical, but how to do this in the everyday real life? So you go to a lab that you just arrive. If quality control procedures, Michael mentioned about accreditation, it exists, it seems okay. But even when it seems okay, you have to be cautious, as we will see. But now let's imagine the most difficult condition. You arrive in a new lab, you don't know anything about the instrument, and most of the staff is out of holidays. There are only some master students that are here to help, to say where the instruments are, where the chemicals are. And as Michael said, we make now a presentation for the labs, taking example in the lab, but it's exactly the same if you have to measure and use instrument in the field, exactly the same. So you have to use balances, pipette, oven, many instruments like this that will not give you the result you are expecting, but that are part of the environment of your lab. Are they regularly checked? Have you access to the logbook of calibration? There should also be, when you have good laboratory practices, a logbook where all the problems are indicated. Every time you have to indicate what the problem is, when it was repaired, how it was repaired. Do you have access to this? So, example, you see, for calibration of a balance. And so ask yourself, every time you have used a balance in a lab or in the field, have you done this before getting your data? Have you calibrated or checked the balance? And who is doing this before you arrived, and how often was that done? And if you do it, how often are you going to do it? And remember also to look, when you use the instrument, the user's manual. For some instrument as simple as a balance, have you ever been checking the user's manual of balance? Because if you have a look, you see, for example, I took this just as an example. I don't make advertisement for that balance or whatever. but you will have 20 or 30 pages of explanations. You see about the workroom, vibration free, free of draft, so on, and also leveling. The balance has to be leveled. And also you see switching on. There are many things that are explained even to switch on a balance, and some tips, recommendation. You see, have you ever looked at the user's manual of your balance? This is something that you have to do. Okay, so you have to calibrate the instrument by yourself. Now I spoke about the balance. Perhaps you have a special section that is taking care of this, and you are very lucky, you have a logbook, everything is going well. But sometimes you have to do it for your instruments, and you have to use a calibration curve and ask yourself, am I in the correct range? I will not detail this, but even for something as simple again as the pH meter, you see, you need to have a calibration curve. I will not detail, but it looks like this. And you have a step to calibrate it. For example, you have to use the buffer 7 first and the buffer 4 secondly only. And after this, there is an impact of the temperature also on the pH meter. So you see, this is what I mentioned about the context. So you have to check everything. And even if all those calibration, I mentioned just about the balance in this case, and for example, a pH meter, but remember, it's the same for the oven, for the pipette you are using, everything has to be calibrated. And even if all those calibration have been made correctly, it's important to have an idea of what range of data you expect. And if you are out of range, check everything. I think this is also what Mohamed has mentioned about one of the results they got that seemed to be strange, and they had to check. This is what Michael said about the cap of the EC meter or pH meter that was not taken out. And I have an example also in my lab, but as I give example, I will not detail this. But it's not always possible to have an idea in advance of what kind of result you will have. And moreover, your instrument can be shifting during the measurement. For example, the temperature in the room is increasing, or the instrument itself is heating. So you need to have internal quality control samples. These are samples for which you know the characteristic that you have to keep a large amount of your lab, and also preparing, storing that kind of sample. There are some special procedures that need to be explained and detailed. But now imagine you have your internal quality control sample. When you are measuring your own sample, you have to introduce one quality control sample, let's say 1 on 10 or from 1 on 10 to 20. This is 5 to 10% control samples that you have to prepare yourself, because there is a lot of samples that you will need. And so you must not buy them. It would be excessively expensive, but you can easily prepare them by yourself. And when you have your data, you generally need to transfer to a computer, because very often you write down your data on a notebook, and take care also to the transcription mistakes. This is a very important factor of mistakes. Very often you have your data, you have checked everything. Okay, the data looks correctly, but after this you enter in the computer, you see 2 or 3. It's so easy to make a mistake in this case. Okay, and remember also that all results are a value plus or minus uncertainty. So when you have your result, you must try to calculate the uncertainty, and you can easily have a look by coefficient of variation or something like this, but immediately have a look. If there's a problem, you must go back and see your set, your bunch of samples, and see what the problem can be, your instrument. And there is not only the internal quality control, but you have also to participate to inter-laboratory comparison. And such comparison can be organized by professional providers, or you can organize yourself with some of your friends or colleagues using the same samples that all of you will analyze. And as Michael mentioned, it's not because a lab is in UK, Australia, or wherever that the results are better. You see, everything depends on these procedures, and this depends on human being, and you don't know. So, as I told you, this is not only what we said is theoretical. It was already mentioned that by honestly, you have to provide good results because it's important. There are impacts on your results. But also the journal, we speak about publications, the journal will ask you more and more often to prove that your data are reliable, because there is a reproducibility crisis, because research is expensive for many reasons. You have to prove your results are good, and the donors will have the same questions. We mentioned about publication, but for me, something that is very interesting is to put your data in a repository, because you can get DOI. That means that your data can be cited and be included in metadata, but you have to include metadata. And this, I think, for me, is a first step to write down, because very often students think also, oh, it's the procedure, material and method is easy to write down. I have done it. No, it's not easy to write, and it's a good exercise to start writing. And as I mentioned also, your data can be cited later by others, and everybody will be able to see that your data can be trusted. So thank you for your attention, and do not hesitate to ask questions and make suggestions of trainings, potential trainings.
Okay, we do have some questions in the chat box. I'll go from reverse order. So Christian, we have something from Maureen Ondayo.
Yeah.
Can we give a highlight on how to ensure quality in the field outside of the laboratory before the samples reach the lab? Elikos and Ariadne might be able to answer this. So correct me if I'm wrong, Elikos or Ariadne. You've both provided links in the chat box to previous reports. Those reports, I suspect, will have details on how the quality was planned into the sampling programs.
Yes, if I can say something. Hello to all. This is excellent, and Michael, this was indeed a very comprehensive and excellent presentation, and also Christian, very, very useful. So about the sampling process, there are quite a few developed methodologies. I wanted to say that many people are afraid of how we estimate the uncertainty, and they have in their mind that it's something very complicated, that we have to go into very complicated statistics. It is not like this. There are methodologies and there are empirical methods. It doesn't have to be expensive, as Christian mentioned, or complicated and something like it is super scientific. Or so this link I provided, it is from the Eurachem site, and there you can find lots of guidance and documents prescribing these methodologies that everybody can follow. And they go from simple ones to more expensive and complicated ones. Also, you have to adapt it in terms of the magnitude of the survey and the area you are examining, of course. But it doesn't have to be complicated, and there is guidance, not only for the analytical part, but also for the sampling part. And of course, we have the example of the big data sets, like the examples provided and guidance from the links that Alekos provided in the chat. And there you can see how these methodologies have worked for these really big data sets, like we go for continental scale geochemical surveys. So this is all from me, and also everybody that has, if anyone has further questions, I am available to ask me if you want something, I wanted to say that my PhD was on sampling uncertainty in my early years. So this is where I'm coming from. And I had the opportunity to work with Mike Ramsey, who was also a past president of SCGH. So there is know-how, and SCGH can provide help to you, and anyone who needs this help is available.
Many thanks, Ariadne.
May
Of course,
I add some
Ariadne
comments as
is
well?
on the European board for SCGH and has provided a lot of energy behind the Early Career Research Network. And this new group will be added to that SCGH network, so you will be connected. Sorry, Elikos, you wanted to say something?
Yes, I would like to add something. It's very important, as Ariadne pointed, the 3 reports that I have uploaded. People should write a quality control report for their data, okay? Because you need validation, as Christian has said, is very important. And also, they should not be afraid to mention mistakes that have been found. In one of the reports, you will find that there are wrong results, and we have reported them, and how we have handled the whole issue. So it is very important for everybody to validate his results with quality control samples that they have, as Christian and you, Michael, have pointed out. And otherwise, if they do not really provide this information, there are nowadays questions about the validity of the data sets that are published.
Did you want to add anything? Thank you, Elikos. Thank you very much for the comments. Christian, did you want to add anything to what Elikos and Ariadne have said on the field sampling, planning that in?
Yeah, there are 2 different issues, because field sampling can also be related to geostatistics. And of course, this is part of the scientific work, and you have to know your field and different things. But as it was said, after this, the quality control is simple. The statistic can be very simple too. You see, just a mean and a standard deviation. The main problem is that you have to change your daily attitude. And this is the most difficult. Buying an instrument, you see, is easy. And I think in your presentation, Michael, you have shown some instrument about redox conditions, and you have written the word isotope, and there were a big instrument, probably very expensive. But you can see that even with a pH meter, you have to take care about how you use it, and even a balance. And this is why I say the daily attitude. This is the most difficult. Who from you has already looked at the user's manual of your balance? Who has made a logbook about the calibration? You see, this is something that you have to do. When you arrive in the lab, you have to check they do it. If they don't do, you have to do by yourself. And if you are in the field, if you are using a balance, if you are use pH meter, if you use things like this, you have also to calibrate them, to check them. And it's in that everyday work that you can get the quality of your set of data. And as it was said after this, having good statistical result that you can use for interpretation. But it's really in the everyday life. And I really encourage you to do the exercise about measuring the egg with your friends or in your lab or whatever, just to estimate the uncertainty in the measurement of an egg. You see, all of you can find an egg. Try to do it. It's not only a discussion by Zoom we have. I hope the next time we will be able to be in the same room and we'll be able to do it, and bringing some pH meter with us, because this is an exercise I wanted to do, to bring 2 pH meter, 2 balance, one calibrated correctly and the other one uncalibrated incorrectly. Give it to you. Okay, now which result is good? Which one can you trust? You have 2 different results. How can you make a conclusion? You see, this is very small things in the everyday life, no cost, a different way of thinking, a different way of behaving. And I would like to encourage you to do this and write down everything, the procedures, because it's where you start the process of writing. You cannot write a very fancy and attractive paper if you have not started at that small level of writing, starting by writing your procedure and convincing the people that your procedure is correct, your statistical procedure is correct, statistical in the lab or geostatistical in the field. Start with very small things and improve little by little. So this is the only thing I wanted to say. It's easy and cheap.
So
daily.
There are measures. So I think the main thing is not to assume that any item of equipment is okay. So even the basic stuff like pipettes and balancers, I have seen where people have made their own check weights, because check weights are very expensive. They've been made from nuts and bolts, but they've been checked with another lab. There will be the metrology labs in countries across Africa. I've generally seen the Bureau of Standards who will have very good equipment. So find a partner who can help you raise the quality of your measurements. There will also be many networks such as the Royal Society of Chemistry, the Food and Agriculture Organization of the UN runs the Global Soil Laboratory Network that Christian mentioned, and they are trying to increase the quality of measurements.
Michael,
may I make another comment, Mike?
Yeah.
Go
on, yeah.
I mean, you mentioned that laboratories always make mistakes and you have to be aware of them. And now the biggest mistake that a lot of people are making is that when they get results from certified laboratories, they think that everything is okay. Now, certification means that they follow a quality control sort of system. So they can go and find out any mistakes that they have made. It does not mean that they can produce good results. It's just that they have all the procedures sort of written, approved, and so on. So be very important, be careful about results. If you don't have a system to check your results, then, personally, I will say that you are not doing your work quite correctly. As Christian has pointed out, and Michael, you have to validate yourselves with your own samples, quality control samples. This is very important, and you must remember it. Thank you.
I think we almost need another session on this because we're getting away from the writing workshops, and maybe we need something on quality measurements. But this was my particular frustration with seeing papers for review quality measurements. I think, Christian, did you want to say something?
Yeah, just a comment, because I see Mohamed mentioning about most labs that are not air-conditioned. Yeah. Bashiro that mentioned about the impact of aerosols, and also Odipo mentioning about true calibration is a serious challenge. But so you see, what you need to know is to write down what you need to do. Is to write down the condition. You have different temperature. So write down the temperature. You have your internal quality control. If one day you have, 20, and the next day you have 22, and you see that the temperature has changed, you know that your result is correct, but it depends on the temperature. So it's a bias, and you must find a way to change that bias. And the same for the question of Bashiro. If you know the aerosol, you have a way to measure. Okay, you just measure it, and you have, in both cases, the external quality control, that is the interlaboratory comparisons. You will perhaps detect it. And this is important for you to know where the problem is coming from, and after this to try to take actions. And the true calibration is a serious challenge, yeah, but again, it depends on the range. Michael has mentioned example where there is the health of the people. So perhaps you need to have very low range and take very great care. But sometimes, let's say for soil fertility or something like this, or soil classification, you can allow yourself to have wider range. But the most important is to have a calculation of the uncertainty. Even if Odipo said it's a challenge to have very precise or very good calibration, but you must know what is the quality of your calibration. If you have a low range or wide range or medium, know the information and use it in the discussion of your paper, because you can have different results. And if you want to prove that your results are valuable, you really have highlighted something new. You can say, we check this, we know that we have not perhaps fully precise result, but the uncertainty we have is low enough to prove that there is a significant difference. So you see, collect the data and use it later for your discussion to prove your data are valuable and can be trusted. This is my comment.
Yeah. I mean, it is, of course, okay to have some uncertainty on the data. Ideally, it's as narrow and reproducible as possible, but it may be impossible to have very tight ranges. It may be too expensive, too time consuming. So it's okay, but as long as that is recorded and presented with data. Yeah. We'll get to Ariadne then. We've got a few questions still in the chat box, and I'm also conscious we haven't answered Maureen's question. So Ariadne, if you want to.
Yes, just a comment to maybe link it back to the writing part of the workshop. So in our paper, I think it is very important in the paper, in the text, in the methodology to mention how we estimate the uncertainty, and then have a comprehensive but brief table with the plus minus of our results and the confidence we have. And of course, we build this in our discussion, and maybe in a supplementary material, we have the detailed data of the estimation of the uncertainty. Maybe this is a good guidance also for the people attending here.
It's wonderful. Yeah, that's great. Thank you. Just going back to Maureen's question, she asked, how do we insert quality into the field? I suspect many people will be doing small scale studies, discrete studies, rather than large baseline studies. So have a look at those reports that are in the chat box. But it could be simple things like collecting duplicate samples in the field. Simple things like that. Ultimately, are you preserving the sample in the right way so that you maintain the integrity of that sample from the point of collection to the point of analysis? And one of the many guidelines that ISO do bring in is actually you report what the time is between collection and the point of analysis, because some analytes will fall out of scope because they're simply not stable, and that's fine, but you just report that it's out of scope. So knowing how you're going to preserve the sample and maintain the integrity would be important. We had Christian, there's a question from a depot for you. Question on reporting values, outcomes. Should they not reflect the accuracy possible? Can you see that?
This is the number of digits, and this is also a big question because it depends on your instruments, and after this, how the things are calculated. So again, I think it's really not difficult, but it needs a special description of how we cannot give a short answer. But first, it starts from the instruments, the number of digits that your instrument can provide, and
they're significant figures. Often, what I see in papers, people will report 74.07% recovery. And you think, well, why do you have 0.07%? What's the significance of that value? So it's often that could be a big discussion in itself.
Yeah, because Excel gives you as many digits as you want. So the people just provide it. But for me, when I review a paper and when I see this, I'm very much, concerned about how the people understand the measurement they have made. So for me, when I see tables like this with excessive number of digits, I carefully check everything, because this means the people are not able to understand what the significant digit means. And if they are not able to understand something so basic, I'm very much afraid for the other data. So I really cross-check all the data when I review a paper. So this is something you have to take care when you write your paper.
It can also come down to a very basic human reaction, is if tables are very busy and have too many decimal points, they're difficult to read. Yeah. And of course, editors do their job on a voluntary basis and are naturally busy people because they're asked to do the job because of their experience, and reviewers are doing it on a voluntary basis. So tidying the paper to make it look presentable and easier to read is going to attract a less annoyance from the reviewers. And if annoyers start getting annoyed, you've got less chance of success, or you will get a longer list of reviewers' comments to deal with. But they might just simply go, oh, they've been lazy, they haven't been tidy, let's just reject it. You know, because reviewers are also very busy. So it's a basic discipline, I think, really, for presentation. We have a question from Bwalia Kalunga in Zambia. Quality data is supposed to be audited by an independent person. What happens if there's no auditor? Could you still attach credibility to the data produced, knowing that the procedures were followed accordingly? I think this is possibly mixing issues. So this is more about quality assurance, the management of the laboratory, perhaps. There should be a manager of the lab who actually should be verifying and checking the data of the analyst. Ideally, you would have an auditor who is independent of the manager, who could check the quality of laboratory measurements. But an individual should be able to present confidence in the data and attach credibility simply through doing the checks that we've described throughout, shouldn't we?
Yeah, in fact, there is a, how to say, confusion between two things. The routine laboratory, where the people produce results for others, they don't know where the sample is coming from, and they have procedures, third party or something like this that check the results. And you are a researcher, you are making your own analysis, and you have to check by yourself. And you see, routine and research work, in this case, is a little bit different. And especially as a researcher, you must be able to do the job for your own data.
I'm sorry, I'd probably I think they should be one and the same. Routine and research should be treated in the same way for quality.
But you cannot ask someone else to check your own data. You see, I mean, it has to be done in the same way, and you have to do by yourself all the job to check, because quality officer will look the calibration of instrument and so on. And in that case, you have to do it by yourself and write down everything. This is what I was meaning, but by yourself, and show to someone else if someone else wants to check.
I think for an individual researcher, if that's a student, they could always show their peers, other students in the research group, or who are maybe a bit more experienced, or ideally the supervisor or the boss.
Yeah,
it's always good to have a second pair of eyes look at data, because very simple mistakes are made in Excel. And to check transcription errors as well. Okay, do we have any more questions? I think Adipo's put in there, true calibration is a serious challenge in laboratories in Africa. Any comments, Christian, on that one?
Yeah, this is what I mentioned before. You must know the quality. It depends on the range. You see, it was Eurachem and some other CTAC was mentioned, but there is a very important concept that is fit for the purpose. Your precision and your accuracy has to fit with your purpose. And so the calibration, the true calibration, you must have a calibration as much as you can and measure the uncertainty on that calibration too. There are different ways to do. And again, it's just to provide the information about how reliable are your data. And it's perhaps enough for what you want to do. You do not always need the highest possible quality for every measure you are doing, but to be a measure, you need to know the uncertainty, a value, a unit, and the uncertainty.
Okay. So quality of measurements has generated quite a lot of discussion, hasn't it? Are there any wider comments for the morning? It can include quality or for writing skills. Any other questions across the group before we close? Perhaps Mohamed, would you like to comment on this discussion about quality measurements? Is that?
Sorry.
Oh. We have in the chat box, so Alfred has responded to.
It's a very nice presentation actually, and this story of measuring the egg is really incredible. I mean, I like it very much. Starting from something very simple. I thought even playing this game with my family here, and it's very important. I have a question, Christian.
Yes.
Of course, you have the reproducibility. What should be done? We usually make, for example, in materials chemistry, we make a material. Of course, if we have time, and we can make, we try to reproduce the results. But when we plot when we have plots with error bars, should it be the error bar on one batch, or should it cover all batches? For example, one given experiment is redone and you make the measurements on the material different batches but prepared in the same way. Should it involve different batches, and also should it include the error bar? Should it include also the accuracy of the instrument you use?
I think in that case, perhaps something just like an ANOVA, using the different, you see, replicates or instrument or something like this as explaining factors would be a solution. But I think if we go in the details, this is why perhaps we would need more deeper trainings on some specific aspect that would not concern everybody, because already measuring the error bars and having an idea of each individual result and uncertainty of each individual result is very valuable. And after this, to make the things, as you said, we have to discuss every individual procedure. But I think ANOVA should be a solution. I'm not sure, we have to look more in detail, but it could be a solution.
Thank you.
We have another question that's appeared in the chat box. Job Isabuk in Kenya is asking, how can you control the error when samples are submitted to a third-party laboratory for analysis?
Wow. If I mean, this is the discussion we had about accreditation, and as we said, accreditation does tell you that they're doing the things in the right way. So they should also have certification or certificate for each parameter to join inter-laboratory comparison organized by different institutions. For example, WEPAL in Wageningen in Netherlands is very famous, and also BPA in France are organizing this. And the lab should be able to prove that it has a certificate for this, and for that parameters, for that specific parameter, indicating that the results are better than blah, 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 blah. But after this, you can, depending on the budget you have, you can challenge the lab by providing several replicates of your sample. But again, you have to take care on the statistical analysis. You need to have enough number of replicates and different types of samples, because, for example, for the pH, if you have samples pH 4 and you test the lab for pH 4, this does not mean that he will be able to provide good result for pH 7 or things like this, you see. So it depends on what is the first party lab and how much is your budget.
So submitting replicates blind to the laboratory, appropriate to the measurement or the type of sample you have. So if you have a high pH or a low pH soil, then having representative soils perhaps. But they could, of course, you could go back to samples you've had analyzed previously and submit those blind. You could even make your own in-house materials. So whether you can make a good soil that you can be sure of being homogenized, you could use that and submit that every time you send away for analyses.
Yeah, but if you have to test the lab, even if it's your own sample, you need to have replicates, because unless free replicates, it's difficult to make statistics, and you need statistical analysis.
May I follow Christian's accreditation? One thing that everybody should remember to do when they are sending labs to commercial laboratories is to sign a contract that any mistakes that are found with your own internal quality control, that then the laboratory is obliged to analyze batches of samples, or if they have made a serious mistake, they are obliged to analyze the whole suite of samples. So you have to be very careful when you send samples to any laboratory, even accredited laboratories or certified laboratories.
No, I'd agree with that. I mean, I run laboratories here at BGS, but we also send samples to contract labs for measurements we can't do. And it's very difficult to get that information from big commercial labs. You have to be quite forceful. I've often seen in papers that I've reviewed, and people come back and say, well, they analyzed it in Germany, so it must be okay. Or they won't give me that information when the reviewer's comments come back. And I've seen that a number of times, and that's not really good enough. They ought to, they should report that information with your data. So whatever controls they're undertaking. So that's often why big commercial contract labs are so cheap, because they simply don't report that data. It would take more effort to change their reporting templates, and maybe they don't do them as frequently as needed. That might be a question to ask. So you could ask, how frequently do they run quality checks on their measurements, perhaps? Ideally, that's all on the website and the information that you sign up to.
But again, Michael, the best thing is to have a signed contract. So, you know, that's sign and seal in a sense. Yeah. So this is the safeguard that they will follow the procedures that you agree. And also when you find mistakes, they are obliged to reanalyze the batches of samples. So this is very important. It's up to you to sort of ensure that there is a contract. Otherwise, they will not do it. And also something that we have been doing for the past few years, we do not like our data to be censored. We like to have uncensored data. Okay, that's the other thing that you should, again, force them to give you the uncensored data, but you must have a contract. Otherwise, they will not follow the procedures that you want.
And that's a very important relationship between the laboratory and the person sending the sample, is managing the expectations and clearly explaining what you want so that the laboratory understands. We often get requests, I have a sediment, I would like it analyzing. Okay, tell us a little bit more about your sample. What would you like it analyzing for? Do you want elemental chemistry, organics? You know, there's that, it can be as basic as that. And often the thought behind asking for quality information or how the data is presented isn't there. Often people sending samples don't know. So there's a responsibility on the laboratories to provide that information and coach the person submitting those samples to avoid a breakdown in the relationship. But equally, if you're not clear about what you want to a laboratory, particularly big contract labs that are charging low prices, they will just give you a number unless you're very clear about what you want and if there's any additional information. So they will have that in the terms of reference. So it's worth checking that. Okay. Yeah, I was going to suggest, Ariadne, great idea is perhaps we've got an ECR session for quality measurements and estimation of uncertainty for future SEGH training sessions. Yeah, I think there's definitely a lot of material here. I'm very conscious of time. I think unless there are any other big burning questions, then maybe we should.
There is a question from Agan Leonard. What about quality control for measurements that are taken in situ? I don't know if you want to answer, Michael.
So you're thinking about field probes, I guess, there. Really, that should come down to how you calibrate the field instruments. So are the field instruments calibrated each day with appropriate calibration measures? So that if that's a pH probe, you have pH solutions that are within date, within an expiry date, and they are covering the pH you expect to measure. So that instrument ought to be calibrated daily. So ensuring that your field instruments are working correctly would be a good place to start. I think unless anyone else has got anything to add to that, that's probably, I think that answers the question. Is that okay for the person asked that? Is that Leonard? Okay, nothing coming back. We'll assume that answer was okay, shall we? Right, many thanks to this lively discussion and all of the questions in the chat box. That's wonderful. And many thanks to Mohamed for opening the lecture. Wonderful advice and tips on writing. Thank you for that. And many thanks to Christian also for measurements. And we've clearly stirred up a lot of interesting discussion here and requirements. And hopefully people get the idea that it's necessary, but it doesn't have to be difficult and expensive. You can always ask people for help. It might be that other laboratories can help you, for example. So this is an open session this morning. This afternoon, we're going to have a closed session just for the writing workshop because we have some group activities. So please remember for that workshop, we have a different Zoom link and we will start in 1 hour from now. So it's 12 o'clock in the UK, we'll start at 1 o'clock, okay, in 1 hour's time. So if you can try and be back on time, please, because we've got quite a busy period of activities to run through. And it's Friday afternoon, so I'm sure everyone wants to be away at a good time. Yeah.
Michael, I'm not sure I have the link for the afternoon session.
Okay, so many thanks also to Andrea Mills, who has been organizing things behind the scenes. Perhaps you can put yourself on camera, Andrea. So Andrea will send the link if just in case anyone is unsure for the afternoon. Okay.
Yes, will.
Great, thanks, Andrea. So this has all worked smoothly because of Andrea.
Thank you,
Andrea. And this afternoon is going to be led by Adipo and Akinaz. I hope that's okay. Yeah, thumbs up.
Thank you. See you.
Smiling, so they're looking positive. Okay, have a good lunch, everyone, and many thanks to everyone else from the other societies that have joined us, and you've helped create a really useful discussion. Okay, take care. Have a good afternoon.
Yeah. Bye-bye.
Bye.